Good evening and welcome to the ENLIGHT online lectures. I am Esther von Richthofen from the University of Göttingen. COVID-19 has brought many unwanted and even disastrous changes to our lives, but it has had some positive side effects. For a start, people are much more interested in science. That is good news for universities. Secondly, video conferencing is now much more widespread than it was before. That makes it easier for universities to bring together experts from different countries for public science lectures such as this one. The European University Network ENLIGHT spans across Europe. It brings together 300,000 learners, and the partners share a deep commitment to their social responsibility. The 9 European universities that founded the network decided to introduce the ENLIGHT online lectures as a new format. Each ENLIGHT online lecture invites experts from the partner universities to share their knowledge, thereby bringing a European perspective to scientific debates. This is the second ENLIGHT lecture, and it has the title New Virus, New Detection Protocol. Our first lecturer is Amanda Sierra. She is a researcher and professor at the Azucaro Basque Center for Neuroscience, where she heads the cell biology lab. Some of her research focuses on controlling the identity and function of microglia. Professor Sierra already published and co-published over 60 studies that were publicized in prestigious specialist literature such as the Journal of Neurosciences. Amanda, it's a pleasure to have you here with us today, and the floor is yours.
So hello, thanks for inviting me. I'm going to share my screen. So my name is Amanda Sierra. I'm a neuroscientist at the Azucaro Basque Center for Neuroscience and the University of the Basque Country. And today I would like to talk to you about how can basic research scientists help during a pandemic emergency. And the answer to these questions is that we can do it by teaming up, by being a group. So we formed this COVID Basque interinstitutional group. You can see all the many people that participated in this project. Here you have a website where you can see the different scientists that participated, clinicians, PhD students, postdocs, PIs, everybody joined together. We developed a protocol that you can find in different languages, and we ended up publishing our results in an open source repository, medRxiv. So this is actually the team. I would like to introduce most of the coordinators to you, myself, a neuroscientist, Daniel Marino, a plant physiologist, who will be joining us later for the questions. The coordinator of the whole team was Hugo Mayor, whose expertise is in biochemistry, and we had other people with expertise in genetics, membrane biophysics, liver, and some microbiologists I would really like to mention here. Xavi Guruseaga, who, while he's still doing his PhD at the University of the Basque Country, he was really instrumental in this project, and it's a pity that he couldn't join us today. So as you can see, we were a group of very heterogeneous people that joined together, and why we, with all these different expertise, why we ended up working in COVID for these last few months. Well, the reason is because our country was in a very bad state. The alarm hit us very early on, mid-March. This is when we had the alarm state, the confinement. And we could see cases doubling up, triplicating every few days. And it was very worrisome. And we were all getting news that they wanted all of our testing kits from the labs. They wanted to see how many PCR machines we have to test for COVID, as we will see later. PCR is one of the main tests that I will discuss later. So we could see that the hospitals were really getting close to this state of collapse, and we decided we had to do something. We couldn't just stay at home. So that's how we joined together and tried to figure out how we could help. So we volunteered and we put together this group. We developed a protocol that allowed to test people without using the big kits and the big machines that are usually used in the hospitals, because hospitals were afraid that they would run out of kits to do this analysis. So we decided to develop this other alternative protocol. It took us around a month until we finally, by mid-April, we actually obtained the certification from the main Spanish Institute of Health, the Instituto de Salud Carlos III. So before I tell you a little bit more about what we did, I'm going to briefly detour a little bit explaining how SARS-CoV attacks the body. And it attacks the body by first, this is the virus, a drawing of the virus. It enters the airways mostly, and it infects lung cells. You see, so what it does, it replicates, it makes up copies of itself inside the cells. And it does it by using the cell machinery. This is a blow up of this cell here, and as you can see, the virus has the RNA and the DNA inside the cell, the virus, and it gets out within the cell, and it makes a lot of copies of the 4 main proteins and genes of this virus: the envelope, the membrane, the spike, and the nucleocapsid. So in the early phase of the infection, when the infection is still growing within the body, what we can do is to detect viral genes and viral proteins. This is what tests try to do in the early phase. Later on, the body develops a response, a defensive response, and this is where the immune system pitches in. So we have all the circulating cells. Here's a blood vessel. We have many circulating cells, and they respond to the virus. So if we have this infected cells, this is going to oversimplify the whole immune response, so I apologize for all the immunologists in the audience. Please be aware that this is an outreach kind of talk. So I know this is not the whole truth of the immune response, but it can give you an idea of how it works. So one of the first things that happens is the lymphocytes produce antibodies. This is a lymphocyte, and it produces these antibodies that are specific for the virus. They cover up. And then T lymphocytes detect these antibodies and especially attach to the infected cells, and they kill them. So if we blow up this interaction, you can see this is the virus and this is the antibody that our body has produced that is specific for this coronavirus. So in the late phase, when our body is already responding, or even for many months afterwards, possibly, what we can detect is the immune response. We can detect our antibodies, the antibodies our body has produced to fight the coronavirus. And this is what late tests actually detect. So if we summarize, in the early phase, we can do a nucleic acid test. This is usually done by using a nasal swab that collects mucus from the back of your throat. And from this sample, we can isolate the nucleic acids from the virus and test using mostly PCR, as I will show you now. And in the late phase, this is the early phase, right? And in the late phase, what we can do is to draw a little bit of blood from the patient or the person and detect whether there are antibodies. So as you can see, both tests are very different. They are complementary, and we need both, probably in different stages of because the early phase, like detection of the viral genes by PCR, what it does is it tells you whether you are infected, whereas in the late phase, you detect whether you have been infected in the past, because your body has already developed an immune response. So it's not one test versus the other, we actually need both. So when we started working on this at the University of the Basque Country, the first thing and the most difficult thing we had to do was to organize the team. And this involved a lot of tasks that are not necessarily related to science. The scientific part I will tell you in the next slide, and it was pretty easy. The non-scientific part was actually very difficult. We had to arrange for sample collection and transport. The collection of the samples was done by Osakidetza, which is the Basque health system, but it had to be transported because this is potentially infectious samples. So it had to be transported in a way that was safe for everybody involved. Of course, we also had to maintain the confidentiality. This was very important because we are not physicians, so we cannot know anything about the names, the last names, the medical history of the patients, and so on. So we had to develop a procedure to maintain the confidentiality. Another important issue, which may seem not so important, but in the end it was very important, was to get access to the campus to all the researchers involved. So on a daily basis, we had to ask for permission for everybody involved. Once the sample arrived to our hands, we of course have to decontaminate it to make sure that all the downstream procedure was done as safe as possible. And indeed, biosafety of the researchers involved was one of the major concerns we had from the very beginning. And so we actually had to move, we had to create a new lab in another building that the University had, the Maria Goyi building. So we had to move all our equipment, all our resources into these new labs and organize even places to go and come back. We had to change our clothes. We had to have a procedure for decontaminating, to wear masks, and so on and so forth. So this was a big, big organization. then it became the easy part for us, which is where we have more expertise, which is the sample analysis and testing that I will show you in the next slide. And in the end, what we had to do is to optimize the protocol. What we had is many samples that came from Osakideta, the Basque health system, right here. And we had to compare their results with our results, and that's how we optimize the protocol. So what we did is actually use a protocol. We didn't really develop anything new. We are not claiming any novelty here, but what we did is to compare different protocols that were available and figure out which ones was the fastest and the most reliable and the cheapest, which was also a concern. So in this stage where you need to analyze viral genes, the first thing you need to do is to isolate the viral RNA. Later on, what you need to do is to detect it. And because there is so little, what we need to do is to amplify it. So to detect it, we need to amplify it. So we tested different combinations of different protocols. I'm not going to go into technical details here, but we used different protocols for RNA isolation and different protocols for amplification and detection that are summarized here in this figure. This is one of the figures of the paper we put in medRxiv. And in the end, we settled for the the cheapest and the easiest to implement, which was also the most reliable. So it was a really quick procedure. We didn't need any kits that were running out of stock in most biotechnical companies. We were completely dependent from all the kits and robots that the hospitals had. So in case the hospital testing system would collapse, we could provide a completely alternative, cheap, fast and reliable method. So I'm going to show you just an example of some data that we obtained. We, for the testing, if you remember, these are the nuclear genes, the viral genes. We focus mostly on the nuclear acid, the N gene, which is what the Centers for Disease Control and Prevention in the USA were using. But we have also used probes for the other genes, and they get very similar results. Usually we use a couple of them in each test to confirm that, to make sure that whether the patient is positive or negative. So as you can see, this is the actual of how it looks at PCR. What we have is different cycles that go on over time. And in each cycle, the fluorescence, the labeling increases. So the final amount of fluorescence that we had at the end of the procedure is proportional to the amount of virus we had at the beginning. So in green, you can see positive samples. We had a very good amplification, so we are sure they are positive. And down here, you don't see anything because it's where we had the negatives. They don't amplify anything. And just to make sure that we had a good sample, that this not amplification was not due to the fact that we didn't have a sample, we always have to amplify a human gene, which in this case is another protein. Doesn't really matter. So this is more or less how the PCR works. We did many experiments to make sure that using two different protocols, doesn't really matter, two different types of probes, we could detect up to 4 and more reliably up to down to 40 copies of the viral gene in each sample, which is very, very, very little. So the technique was very sensitive, very specific, and very reliable. And in fact, we validated 100 samples, over 100 samples from Osakideta. And as you can see, we have the match between our results and the results is very high. We have very few false positives and false negatives, which is very good. So pretty much we were obtaining the same results as they were. So later on, what we did is to try to improve this for mass detection. And the way they did this, and again, this is not our idea, other people have also done this, is by pooling samples. Instead of analyzing sample 1, 2, 3, 4, 5, you can pull 10 samples together and analyze them together. And if you get a positive, means that one of them, at least one of them is positive, and then you can identify each one of those individually. But if not, you go in groups of 10, which really accelerates the testing a lot. So this, we compared to validate whether this pooling was good enough or not. We compared the number of copies of the viral genes in undiluted samples and in a 1 to 10 dilution. So this is a 10% less. And as you can see, there's a very good correlation between expected and we actually got. So we can really detect very well the virus even when we pull together 10 samples. We had a few samples that were positive when they were analyzed individually, but when we pulled them together, they were not positive any longer. You can think that if the positive sample had actually very low levels of the virus, if you diluted 1 to 10, you may not be able to detect it, which is true. It did happen in a few samples. But this is very far from the infectious range that has been considered in recent papers from experts in the field. So we think this protocol can be used really for mass detection. If you quantify people in groups of 10, and whenever you find a pool that is positive, then you identify individually which one is infected. So just to summarize, this is what I'm going to let you know what we have achieved and what we have not. So we have developed this extraction-free protocol. We don't need to use kits. It's cheap, it's fast, and it can be applied in many other circumstances. This, for instance, is currently being used, or at least tried to be used in Kenya, because this testing is expensive and our protocol is very simple. We did try to test different groups at risk that were not being analyzed at the hospitals at that time, like firemen, policemen, and healthcare. But there were some political issues, and I don't want to discuss because this is not our area of expertise, and this is not our decision to make. But we couldn't do it. We were able to do it, but we couldn't do it. What we did is we participated in an epidemiological study organized by the Carlos III Institution in Madrid, where we analyzed over 6,000 samples. So, but in the end, what we think is best is that we were able to engage the public and politicians in a very open discussion about the importance of testing to develop data-based policies. At this time, confinement was very strict, and many people believe that we just didn't have enough information about whether the confinement was doing a good job, it was enough, it was not enough. So we needed more data, and this is how scientists actually make decisions based on data. So we needed more data, and we still need more data. We need more tests, more serological tests, more PCR tests, in order to make sure how is this pandemic evolving. And this actually caused a lot of stir in the literature. The PCR tests from the university were were discussed openly. So we think this was actually a very important part of one of the most important achievements of all these procedures that we started. And this is possibly one of the consequences of us making all this noise, creating all this necessity for explaining why it was necessary to really test more people. And this is the number of, in the Basque Country, in the first few months, number of cases, how it evolved. This drop is because they changed the way they quantify it in Spain, so it's not relevant. What really matters, as you can see, is that there was a sudden increase in the number of tests performed in the Basque Country. And this really coincided with the time point where we were developing the protocol and talking in the news about the importance of developing the protocol, and also overlap with our study. So I think we were in part helping to convince politicians and the public that these tests were really necessary. So I'm just going to finish by sort of reflecting on how important basic science is. We usually don't see basic science. It's like the hidden part of an iceberg, like Fernando Cosillo, the head of the Basque Foundation for Science, Seeker Basques, used to say. We don't see it, but it's there. We are there to train people. We are there for solving problems. We are there for addressing the needs of the society, even though they may not seem evident at the time. So it's very important that basic science keeps going on, even if it's not really obvious. So thank you very much for your attention, and I'll be happy to take any questions.
Amanda, thank you very, very much for this insightful talk. I can't even begin to imagine how difficult it must have been getting this group put together. It's amazing how quickly it was manifested, and I'm sure that a lot of work which was planned for this period is actually still lying on your desks, and it's something you can only come back to in many months' time. But thank you very, very much for your efforts firstly, and also for describing them here. It's really fascinating. I would suggest that we start with the second lecture, and then we collect all questions and put them to both you and Tomas at the end. So now, after this fascinating first lecture, we move on to our second lecture. That is Tomas Szemes from Bratislava. He is a researcher and developer at the Department of Microbiology and Faculty of Natural Sciences at Comenius University of Bratislava. He is an expert in the field of genomics and deals with the analysis of genomes. So genomes are the full genetic information of living organisms, such as viruses. His work is frequently published in specialist literature, such as the International Journal for Molecular Sciences or the European Journal for Human Genetics. He and his team helped develop the COVID-19 kits for diagnostic purposes and are now used in routine testing. So, Tomas, thank you very, very much for joining us, and I'm very much looking forward to your talk.
Thank you very much for inviting me. So let's start my talk, which will be very similar to Amanda, because we did a very similar thing, actually. But I will focus more on how we got into this topic, because it was slightly different than what Amanda was describing. Let me first introduce myself. I am a molecular biologist, and I work now at our new science park of our university, Comenius University. And I focus for most of my career on genomics and so-called bioinformatics. What is a genome? Genome is a complete genetic information of a living organism. And it's made of genetic code written in 4 different letters in the structure of DNA or eventually RNA in case of some viruses. So reading DNA is what I do, and it's called also sequencing. So determining DNA sequence, so sequencing. Genomes. Genomes are quite variable in size and of course in structure. For humans, for us, it's made of 3 billion letters divided into chromosomes, and actually in each of our cells we have 2 copies of these 3 million letters, so it's quite large. For example, compared to bacteria, where the genomes are only in the size around 2 to 6 million letters, and much larger compared to viruses, which tend to have relatively small genomes, even coming down to around few thousand letters. And when we talk about viruses, they are very variable also in their structure, and we know RNA viruses and we know also DNA viruses, which have a difference in the molecule that bears this genome. And actually sequencing is the given method that is used to identify new viruses, and also to study how they are related to other known viruses, of which we already studied quite a lot. And of course, the novel coronavirus that we are talking about today, which has the name SARS-CoV-2, was also discovered by sequencing the first isolates around January. The size of the genome is around 30,000 letters of RNA, and it contains about 15 protein coding genes, which are responsible for for all the features of this virus. So how do we do sequencing currently? There are different methods, even some newer ones, but we do it. It's not that simple to read the DNA. You have to have a special molecular method for which authors, one of the authors received his Nobel Prize, Frederick Sanger, but we now do it with a so-called second generation sequencing or massively parallel sequencing. So the DNA molecule that you can see on the left side, upper left side, is the double helix structure, which contains those 4 letters, which represent A for adenine, T for thymine, guanine, and cytosine. The sequencing is basically cutting the DNA into small pieces, sticking it to the glass slide, multiplying the molecules on this slide, and then using enzymes that are present in the cell in some special method to read this DNA. And we are also using so-called fluorescent labels to distinguish between each letter. With sequencing like this, we can read very short pieces of DNA, only around 50 up to maybe around 150 letters, which wouldn't be enough to read the whole virus in the case of SARS-CoV. So we do it by applying so-called bioinformatics, and that is helping complex algorithms that are put into so-called pipelines, which you can see on the bottom left side, help us to take each sequence, compare with some so-called reference genome, and put it so that it helps us to read the whole DNA or RNA of the organism. And that way we can reconstruct known genomes and find differences, or even in a slightly different way, we can find out how newly not yet known genome looks like. And that was the case of SARS-CoV-2. Maybe I will return to 2 slides back, where you can see a picture of known genomes of the new coronavirus, in which you see that there are some, different colors with different letter designation. Can you see it on the right side? And this, so this is a map of known genomes of novel coronavirus that were acquired around the world using sequencing, which helps us understand how this virus changes, because there are some small tiny changes accumulating in the genome, and those changes help us first to understand difference and maybe track viruses regarding their source, but also they help us to study such thing as severity of the disease between different countries, regions, or or continents. Okay, so let me continue with this slide, which is data from yesterday, published on GSAID database, where you can see that there are already 10 million known cases of coronavirus disease individuals, and we see that the global death rate is 500,000, which is around 5% mortality. I have to say that the corona pandemic is not yet over, and it actually is getting slightly worse. We have new record in newly identified cases each week, so the situation is still ongoing and severe. GSAID, which I mentioned in the previous slide, is also the database where the newly sequenced coronaviruses are gathered together, and then based on some bioinformatic methods, they're studied, and they can help us in basically monitoring which strains got to which place and which are tending to be more prevalent in different countries around the world. At the bottom side, you can see the genome, so the whole genetic information of the virus stretched to a line. And you above that, you can see a graph that shows you in which places in the genome we see differences, so changes in the letters. And they're, as I mentioned, important to study their relationship and their sources and how they tend to evolve. Let's go to the situation in Slovakia. Of course, the first alarming information came from China in January, and then we were shocked by massive rise of cases in Italy and Spain. This was already at the end of February, and but we have had a special situation in Slovakia. We had parliamentary elections on 29th of February, so the government didn't really pay a lot of attention to coronavirus. And the voice of whistleblowing scientists, virologists, was largely ignored. There was almost no reaction of government by that time. It all started basically in March when crisis was declared, and we discovered that we have no strategic reserves of protective gear for healthcare personnel, but also what we didn't have was molecular test to test for presence of virus in suspect patients. So the reaction was that schools and state institutions, companies, malls, sporting events, other public events were all banned, closed, and we all had home office and children at home. And also there were additional measures taken, such as we had very early mandatory use of face mask outside of your home, and also state quarantine was obligatory for all incoming citizens since 15th of March, I think. Interestingly, the situation didn't get that bad in our country. So as of yesterday, we have only 1.6 thousand cases and only 28 deaths which were connected with coronavirus disease, so COVID-19. Let me continue with how we got into this topic of well, dealing with coronavirus. As I mentioned, my work is to sequence DNA or RNA, depending on a given topic, and to sequence genomes. So we already had an ongoing collaboration with a top virologist group in Slovakia at Slovak Academy of Sciences. So we were the first ones to sequence our first patients, the first isolates from Slovakia. And we did it in a slightly different way than usually is done. So we had these viruses grown on so-called cell cultures. So they were grown in a glass tube in a special, highly safe laboratory for working with viruses. And then after that, the RNA was isolated and it was rewritten into DNA to make it safe. That DNA was then transferred into our laboratory, and we reread the DNA and we put it together using bioinformatics. And that allows us to identify the source of first infections happening in Slovakia. And we learned that one of the virus came from Wuhan. Two other isolates were carried from Paris, and one of the isolates was from Groningen. And these were isolates from March, and we came to this result by the end of March. But we have in the country, we had this completely different problem with the supply of fast testing method for presence of virus. So the sequencing is really the best way to identify a new virus, because you read the whole DNA or RNA, but it's very slow and it's not suitable because it's very costly. So for comparing, it takes 1 week to get your result. So it's not suitable for handling pandemic. And one sample typically costs around 1,000 euros. So that's also not feasible for a healthcare system. To monitor a spread of pandemic in a country, you need a fast and cheap and of course reliable method. And that was what Amanda was also talking about. That is a method based on so-called real-time qPCR analysis, which is fast, sensitive, and cheap. But we didn't have access to kits because, for example, some supplies were cut due to countries stopping exporting these kits. They needed it for their own use. Therefore, we started an initiative, a couple of colleagues from different institutions and even some companies. We put heads together and decided that we would create our own kit. This started on March 19. And also what we did, the reading of the genomes helped us in a slight redesign of WHO approved protocol. So we made slight changes, let's say, to improve sensitivity. Our role in development of this homemade qPCR test was to select the proper combination of key enzyme mixes, also to determine the proper volumes of each of these so-called reactants. We were working a lot on optimizing reaction conditions to make it as sensitive and specific as possible. And we, of course, had to check how it works with real-life samples, which aren't always the same as your perfect samples that you can order from companies. And at the end, we had to determine the sensitivity of our so-called assay or the test. As mentioned, this was a partnership. We partnered with a top virologist group from Slovak Academy of Sciences. Also us worked at the science park of Comenius University. And we had a small biotech SME company called Multiplex DX, which was responsible for designing those so-called primers that were mentioned in previous lecture. And also they had the manufacturer for producing those. We were also lucky that a private company called ESET, which you may know, was funding this whole effort. And then we had some experienced biotech companies that helped us get CE-IVD certification for our kit, which is important for diagnostic use very quickly. And the result was that at the end of, in the middle of May, we provided 100,000 tests for the country. So this was a gift. This is how the kit, how this test in the form of a box looks like. And this is what is then sent to laboratories to use for testing of coronavirus. For us, this is not the end of the story, not just because the pandemic isn't over, but also because it's a very interesting biological problem which needs to be studied further. We would like to focus more on the host, so on the humans. We would like to study how different genetic variants in humans influence how the severity or incidence of the disease or the mortality rate changes. And we would also like to contribute with more genomes to help in designing of better vaccines for the future. That would be all from my side, and I'm happy to take questions from you. Thank you.
Tomas, thank you very, very much. And also, it's nice to hear that it's not the end of our story, not just because the virus is still with us, but also because it has generated so much more options and opportunities for further research, which is what science is about. It's not staying still, but always finding new questions to answer. Thank you very much. That was incredibly interesting again. And I myself understand a lot more now about the testing. I would like to introduce a researcher who joined us for the discussion. He's not going to give a talk, but he will be there to answer questions. He's from Amanda's team. Hello, Daniel Marino. Daniel is a scientist from the University of the Basque Country and the Basque Foundation. He works on plant physiology and molecular biology in the Department of Plant Biology and Ecology. His research is focused on studying the genetic basis of plants' nutrients, use efficiency in the context of reaching a more sustainable agriculture. You must forgive me, I'm a historian by training, so I'm still I'm learning more than most at this point. We've had a few very interesting questions. I would like to ask the first one to, I think, all three of you. The first one is about the reliability of the tests. If you get the result, how sure can you be that they're accurate? I mean, Tomas, you talked about how costly the tests were and how we had to find much cheaper solutions to conduct much better tests. So how much can we still rely on their accuracy, is I presume what that question is about. And I'd like to also put it to Amanda and your team, of course.
Okay, so one part of so-called validation of a new test is to determine what is the so-called limit of detection. So how much virus can you detect in your reaction? And that's what we did. So we determined it similar way as was presented by Amanda, by basically putting a known number of copies of the virus into reaction. And what we came to was that even at the amount of 2 molecules of the virus in the reaction, so 2 viruses, you can detect it with 95% confidence.
Okay.
So that's our limit of detection as was certified.
Thank you.
Yeah, I think what's important is to validate. So every technique has limitations, but so the question of non-specific detection and false positives is very important, mostly with serological tests, because sometimes they do not really pick up the antibodies. But PCR is super sensitive, super reliable, and as Tomas is saying, it really has the capacity to detect very, very low numbers of copies, actually much lower than it's actually infectious. So in this case, in our case, what we did is to compare our results with results already validated by the local system. So we had 95% of match between our results and the results. And the thing is that this is not, it's not a black or white. You have more copies or you have fewer copies. And when you establish the limit of, is 1 copy of the virus really infectious, or is 2, or you need 100, right? So because it's in the low range is where you can have some sort of discrepancy in whether you interpret that as a positive or as a negative. But in the end, those low numbers are probably not very important because they are probably coming from people who is not infectious. I don't know, Danny, would you like to add anything?
I think that more or less what you said, what we think is that if someone is infectious, the probability to have a positive result is almost 100%.
Really? Oh.
So when very, very low copies or very low virus load is present in someone's body, there we can have a doubt. And probably if that person has some symptoms in the future, the test will be repeated. So I think it's really, it would be rare to skip someone that can be infectious with the PCR.
Yeah. Yeah, actually, in the protocol that we developed, we did, so we have clear positives, clear negatives, and some doubtful samples that our recommendation is that in a few days they should be retested again.
Okay.
See if they will evolve into positive or into negative.
Maybe I can add, the detection is divided basically into 3 steps. First is collecting the sample, which is putting a stick into nose and mouth. The second is isolation of the RNA, sometimes skipped, as was mentioned by Amanda. And the third is this real-time PCR test. This is basically 100% sensitive and very specific. The problem is with the first step. So the biology of the virus is such, as was presented by Amanda in one of the slides, that it actually drops in amount in your nose and or in your mouth as it is going further into the infection. So it's basically sort of moving into your lungs. But at that point, when it's in lungs, it may also be not present in your neck or in your mouth or in your nose. Okay, and so that's when the tests fail, even though the patient is positive. But of course, no one is going to put or take sample from your lungs when you have some symptoms that tend to look like you are going to fail with your lungs. So that's a complication, but it's due to the biology of the virus.
Okay. But I presume that those people would then also be less infectious, or are you as infectious as before if it has moved down to your lungs?
I think we don't know much about the progression of the disease yet. Most of these guidelines have been developed over the last few weeks or months. So we don't know, in the future we will be able to know how much viral load do we need to detect in the upper respiratory tract, how much in the lungs, how much in the saliva, to make sure, to establish whether the person is infectious or not. But this is still a little early.
Okay. The big problem is with a relatively large group of people who are infected, but they have absolutely no symptoms or so light that they don't even discover it. So we know about patients that are positive consecutively for 5 weeks or even more. So they have positive qPCR tests for 5 weeks. They're infectious, but they have absolutely no symptoms, but they have to remain in a home quarantine. So this is a big problem, and one thing that we don't understand fully yet. So it has to do something with the immune response or lack of it, but at the same time not causing harm to the individual, which is very interesting.
What's absolutely fascinating for me as someone coming outside from your field is, for the first time in my life, I'm actually seeing how medical science works, and how in a very short space of time, we are seeing how research in medicine works, how you are advancing, getting your findings. I mean, with any other, even the HIV pandemic, the timeframes were much, much longer, but it's a kind of the whole world is looking over your shoulder while you are trying to do the research. So I presume that must be quite challenging.
I think if I may have a comment?
Yeah, please.
I think it's interesting because you guys are seeing science done in real time.
Yes, exactly.
So you are seeing things that may not seem very solid maybe, but for us it's completely logic. Like you have one study, then another study that says the first one was wrong. And this is how science actually evolves. This is how we actually improve. I understand for the lay audience, this may seem like, oh, scientists don't have a clue what they're doing. But this is how actually science works.
Yeah.
So I think it's interesting that this is somehow broadcasted in public to see the, you know, the little insight of how science actually works. And it works by error, trial and error. We try something, we interpret the data using a hypothesis, and then new data come up when we say, oh, the interpretation was wrong, now we have to believe something else. So this is how science works.
Yeah, absolutely.
But we have to say that it's fast forward space now, because at the beginning it didn't see we have too much time to waste. So also you can see some errors happening.
Yeah.
For example, CDC made some. They had situation with so-called positive control. They had contamination, so they weren't able to produce their kits and provide them to their institutes. And similar things happened around the world, even in well-established companies. So it was a pushing situation.
Thank you very much. And we'll move on to our second question so that we can actually address all three. The second question is basic science and politics. How surprised were you about the political debate? Have you ever experienced something like this before? I mean, of course, please don't I think we shouldn't misunderstand this question for you to actually take a political stance and saying, you know, which approach by which politicians were right or wrong. But I'm taking this question to mean just how you feel the political debate has affected the whole.
I have to say it was very, very difficult. It was very difficult for us because we were just volunteering, you know, we were trying to give our best to help the society. Yeah. And all of a sudden we came under the fire for reasons we still don't understand. So it's like they try to create a fight, but in the media, in public. Yeah. So we were like really shocked. It was, you have to understand that we had over 40 people. There were PhD students, we had postdocs, we had people in different stages of their career who just wanted to help. And it was so difficult for them all of a sudden to feel like, but why are they saying these nasty things? Like, because they were saying, they were trying to discredit our work somehow, right? So it was very difficult to keep them working and motivated. So people wouldn't feel like, but why is this? Why are we being attacked? So that was also part of the learning curve here. Well, how was for you, Danny?
But that's the problem that, you know, nowadays, I mean, with all the media attention on you, each single media outlet wants to get the maximum clicks, and you don't get the maximum clicks by just reporting things are progressing as they should be. You kind of need something contentious. So,
but it was not the media. The media was supportive.
Oh, okay. It was the politicians.
It was actually the politicians who,
really?
Yeah. Well, I'm not exactly sure exactly who. I don't know if it was the local. It doesn't matter. But it was a political decision that we shouldn't do tests.
Okay.
And they did it by somehow announcing in public that even if we had this validation, that it was not good enough, and that we were only for research. What we did was only research. You know, it was like trying to dismiss everything we had done. So that was very hard.
That's tough. Tomas, have you had a similar experience?
I think we had relatively good support. Even our test was introduced by Prime Minister together with other politicians. So there was a support. There were actually some attacks after introducing our kit and trying to discredit, but it wasn't a powerful voice.
Also from
and we had a really good coverage in media, so
okay. Good. Well, it's wonderful.
I was just gonna say that we really had a good coverage in media and support, but I feel that the administrative bureaucracy, this bureaucratic engine, was not able to adapt quick to this emergency situation. As we did, the scientists, that we changed what we were doing from one day to the other, but all these steps that are needed, like to change something so we could start doing this diagnosis. So that could be one reason. This bureaucratic thing that was not able to adapt fast enough,
Yeah.
or something like that. But
well, you're right.
It's a question.
Bureaucracies aren't known for being very adaptable. Quite the contrary, in fact. But it is very interesting to have those very different experiences. Here in Germany, we have a situation where we are actually approaching an election year. So each and every politician is kind of seeing this as an opportunity to put themselves forward as a candidate for leading the country. So that's putting us in an interesting situation. The final question I would like to ask is a question: how did you organize your research during lockdown? And of course, it can't have been easy working in the laboratories, wearing masks and keeping distance all the time. How did you manage that?
I personally, we worked 24/7 for many weeks, because we did double time. We worked in the COVID project, and at the same time tried to still work in our own projects with our own PhD students, the thesis, the papers, the writing, the grants, everything at the same time. So it was, we had 2 jobs at the same time. So it was really crazy, yeah. Really crazy. And plus the family, some of us have small children at home. Yeah. So this really was a test.
And I think also she's asking on how we manage the security, or the biosecurity thing. So in our case, we used as much protective personal equipment as we had. I mean, much than it was really needed for doing a PCR. So we were wearing clothes specific for the lab, disposable lab coats, masks, and it was difficult to find them at that point. And even in the beginning, the animal facility of our university gave us all that material. And also we were keeping distance. We were working in small groups and trying to be in different benches and trying as much as possible to keep distance. Mostly not to get infected ones to the others, because if only one person, we were thinking that if just one person at some point was positive, the lab was closed, because what they were doing at that moment was all people in contact. They were telling you, okay, go home, stay there for 2 weeks. And so that would mean that the lab was closed. So we were taking much care about this protective, using the protective equipment.
Thank you for sharing your experiences here. Tomas, what were your experiences?
So our university really sent students home and all the employees home for home office work even before government announced basically lockdown. So the university, including our science park, was closed down, but we were granted to access as we were working on coronavirus. And so we were only few people in the building, and it was actually quite good, because even traveling to work took the least time that ever happened in the history. So empty streets, roads. Actually, on this topic, on coronavirus, we had a very effective work. But of course, the ongoing projects that we have, they were stopped. Especially, we do some research on cancer patients, and all the healthcare for cancer patients was stopped, and it really took its toll. For example, there was some data published yesterday that death rate due to other reasons than coronavirus actually doubled in April and in March and April in Europe. So definitely coronavirus has its other victims as well, which do not have to do anything with the infection. So we have to be careful about this as well if we get into the second wave in September.
That is very true. I mean, all your efforts are now focused on one thing, and not just yours, but everyone's in your field. Thank you very, very much for sharing. I will just check. I can't see any other questions that arose in the chat. So I would like to thank all of you very much again for participating and making the second ENLIGHT online lecture a truly special and truly fascinating event. Thank you for taking your time despite the 24/7 days that lie behind you. And it's just my hope that the coming months will be different and will give you a bit more breathing space. I very much look forward to the ENLIGHT online lectures beginning again in the fall, hopefully. And I would like to thank you for joining in and listening in, and I wish you a very pleasant evening. Thank you very much.
Thank you.
Thank you very much. Bye-bye.
Bye-bye.